Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to our 5th lesson on genetics. Last time, we learned about the stages of meiosis and the formation of haploids. Today, we are going to learn how the sex of an organism is determined by its chromosomes. We will also learn why the common fruit fly is an ideal experimental animal for genetic research. Are you ready, students? Then let us begin. We have already learned some facts about sex determination in previous grades. We know that sex is determined by the X and Y chromosomes. Females have 2 X chromosomes and are called the homogametic sex, while males have an X and a Y chromosome and are called the heterogametic sex. In addition, both men and women have 22 pairs of non-sex chromosomes called autosomes. In any mammal mating, including that of humans, the predictable ratio of males to females is 1:1. Although it is the Y chromosome that determines an organism's sex, only one gene on the chromosome, called the SRY gene, determines the formation of testes. In the earliest stages of embryonic development, a region called the urogenital ridge develops into something called a bipotential gonad. This structure has the potential to develop into either ovaries or testes. When the SRY gene is activated, the bipotential gonad develops into testes, so the embryo becomes male. If there is no SRY gene in the chromosome, or if the gene is not active, then the other genes on the X chromosome and autosomes cause the bipotential gonad to become an ovary. In this way, the embryo becomes female. Sometimes an individual may be born with an abnormal number of chromosomes. Students, why do you think that is? Form into groups and take a few minutes to discuss this question. Think particularly about what you have learned of the process of meiosis and how this might affect chromosomal distribution.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. An individual may be born with an abnormal number of chromosomes because of a problem during the process of meiosis. Sometimes the sex chromosomes do not decouple properly. This means that the chromosomes do not segregate into the gametes. When the abnormal gametes fuse with normal gametes, the abnormal chromosome numbers can result in a number of issues. Organisms with only one X chromosome, denoted like this, develop into females with slightly masculine features. This is called Turner syndrome. In the same way, organisms with two X chromosomes and one Y chromosome develop into males with slightly female features. This is called Klinefelter syndrome. Individuals who are born with too many or too few chromosomes are sterile. This means that they are incapable of having children of their own. So far, we have discussed the mammalian method of sexual determination. It may be surprising for you to hear that there are a number of other methods completely different from our own. As an example, for bird species, it is the females who are heterogametic and the males who are homogametic. However, birds do not have X and Y chromosomes like us. Bird chromosomes are W and Z. So, female birds have WZ chromosomes and males have ZZ chromosomes. In some reptiles, such as alligators, have their sex determined by the temperatures at which the eggs are incubated. Males are produced if the eggs are incubated at around 31.6 degrees Celsius, and females are produced if the incubation is significantly warmer or colder. Some species, like earthworms, have no system at all to determine sex. They are hermaphroditic, meaning that they have both sets of sex organs. It is time for our second activity, everyone. We have learned a lot about sex determination systems. Let us do a quiz to see what you have managed to remember. Please fill in the blank spaces in the following sentences. Do your best, and I will see you in a few minutes.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again. Let us go over your answers together. Males are heterogametic because they have two different sex chromosomes. The SRY gene is responsible for the formation of the testes. The urogenital ridge develops into the bipotential gonad. If there is a problem with the disjunction of sex chromosomes during meiosis, they do not segregate properly into the gametes. Female birds have WZ sex chromosomes, and male birds have ZZ sex chromosomes. Earthworms have both sets of genitals, so they are hermaphroditic. Good work, students. Now, let us shift our focus a bit to the subject of experimentation. If you look in your textbook, you will find that a large number of experiments involving gender and genetics are carried out involving fruit flies. Why do scientists like to use fruit flies for experiments? Take 2 minutes to jot down your best guesses.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello once more. Let us compare our answers. Fruit flies are small animals with a short life cycle. This allows scientists to study many generations in a short period of time. They are cheaply and easily bred and kept in large numbers. They have 4 pairs of chromosomes per cell, and the chromosomes are large and easy to see. Scientists can use staining techniques, which reveal dark bands that correspond to particular genes. I am confident that you were able to come up with some of these answers yourselves. Great work today. In this lesson, we have learned about sex determination and fruit fly experimentation. Until next time, thank you, teacher. Thank you, students.